So today we are going to take a look at some classic experiments that led to the discovery of DNA as our genetic material. We are going to be looking at a timeline of the history of DNA's discovery, and we are going to be looking at the big three, if you will. Now, here in the Detroit area, when we say the big three, of course, we're always talking about what's it called? Ford, GM, and Chrysler. Those are the big three here in Detroit. But in the history of the DNA, the big three refer to the three fundamental experiments which established DNA as the source of our genetic code. And that is the transformation experiment, the Avery's experiment, and the blender experiments. And I love that name. Like they're like referred to as the blender experiments, which is kind of cool. All right, so as we go through the history of DNA, we are going to look at a bunch of different people. And in your notes, ladies and gentlemen, we start off with a timeline. So, our first scientist that we want to take a look at is a dude by the name of Frederick Miescher, followed by Albert Kossel. I can't pronounce half of these names, and Phoebius Levine. These three guys, for the most part, we are not going to talk about any of their experiments because these three really did more work with the macromolecules that we remember from first semester. Okay, so remember there are the four basic macromolecules that build all living things. They are carbohydrates, lipids, proteins, and nucleic acids. These three guys mostly did their work with the macromolecules. Starting to look at actual genetic material, we have Frederick Griffith. We've talked a lot about him this week so far. Oswald Avery, we've also talked about him. Have not talked about Erwin Chargaff, but we will talk about him a little bit more next week. Martha Chase and Alfred Hershey. This is the Hershey-Chase experiment. Yes, we've talked about them this week. So Griffith, Avery, Chase, and Hershey, those are the big three, right? And then next week, we'll take a look at Rosalind Franklin, Wilkins, and Watson. Oops, let me back up. And Watson and Crick. So Chargaff, Franklin, Watson, Crick, and Wilson, those guys are we're going to focus on next week. But for right now, we are taking a look at Griffith, Avery, and the team of Chase and Hershey, excuse me. All right, so on your timeline, our first guy on the timeline is our buddy here, Friedrich Miescher. I can't pronounce that. So all the way back in 1869, remember, at this point, Mendel has already done his work. We know that things are inherited. We just don't know how it happens, like at all. We know that there are these factors. We know that there are these alleles. We know that stuff gets inherited, but we're not exactly sure how. So what Miescher does is he discovers a phosphate-rich material, which he calls nuclein, in 1869. And he finds this in the nuclei of white blood cells that he found, I know, disgusting, on the pus bandages of his patients. So he concluded that it must be present in cells. For those of you who don't know, basically all pus really is, I know, gross, is basically your white blood cells when they explode. Because a white blood cell, its function in the body is to literally go around and eat bacteria and viruses. And kind of like you and me, if you eat too much, you kind of go, and that's what pus is. So kind of gross, but it's how he discovered nuclein. Kossel comes along in 1878, and he actually looks at this nuclein material and decides that it's not this crazy combination of kind of gross stuff that's in pus. He actually is able to isolate nucleic acids out of it. So if you recall from our four macromolecules, we talked carbohydrates, we talked lipids, we talked proteins. Kossel is the guy that actually isolates the nucleic acids, and he separates them from the protein. Prior to Kossel, we thought that proteins and nucleic acids were kind of like a combo deal. They're not. He also discovers that there's basically 5 nucleic acids, which really makes the nucleic acids kind of simple chemically. And we'll talk about these 5 more this week, or next week, excuse me. But there are basically 5 different subunits of nucleic acids. Okay, pretty simple stuff. And again, if you remember from our macromolecule discussion earlier, there are two basic types of nucleic acid. There is the DNA, and then there is RNA. He didn't get that far. He just figured out that there's 5 different types. All right, cool. So after this guy, along comes Levine, and that's in 1919 at the height of the Spanish influenza pandemic. And he discovers that each subunit is basically made up of a sugar, a base, and a phosphate. Again, we talked about this. We talked about what's it called? The macromolecules back during first semester. All right, so these are the basic subunits of this nucleic acid: phosphate, sugar, nitrogen base. All right, so finally, something exciting. Frederick Griffin, 1928. He's working on a cure for pneumonia. Keep in mind, he has survived the Spanish influenza pandemic of 1918. So this is 10 years later, and they're still working on a vaccine for the flu. He's working on a vaccine for the flu when he kind of accidentally discovers this transforming principle, is what he calls it. And how he does this is he's working with 2 strains of pneumonia bacteria. One, which he refers to as the rough bacteria. It's literally because when you look at it under a microscope, the edge of the bacteria has these rough edges. Like, it's just, so he just called it R-type because it looked rough. And he also is working with a smooth version of the bacteria. Again, when you look at it under a microscope, it appears to be smooth. A nice smooth edge. Now, the thing is, the rough strain is pretty innocuous. It doesn't really do a whole lot. Doesn't kill anybody, doesn't even barely make you sick. The smooth version, however, is deadly. And he injects this stuff into mice to see if he can come up with a vaccine. But this is what he actually comes up with, guys. So again, we're looking at Griffin's experiment. He starts off, and again, this is under the big 3 in your notes, under transformation. This is his experiment. Test number 1, he uses the rough strain, injects it into live mice. So he injected the rough strain. It's not deadly. And big surprise, the mouse lives. No shocker there. His second test, he took the smooth strain, which again was the very virulent, deadly version of the strain, injected into the mice, and big surprise, the mouse died. In his third test, he takes some of the smooth strain bacteria, which he's heated, right? So he has heated it, and we know that heating destroys proteins. That goes back to denaturing. We talked about that during first semester, during macromolecules. So he heat kills the smooth strain, injects it into the mice, and big surprise, they live because you've just injected them with some dead bacteria. Wow. All right, cool. But the interesting one happened on the 4th experiment. During his 4th experiment, what he did was he took live rough strain, which we know is not deadly. He took heat-killed smooth strain, which we also know is not deadly, injected it into a mouse, and the mouse died. That was an interesting outcome because you took 2 non-deadly things, you put them together, and somehow you ended up killing these mice. The rough strain was therefore somehow transformed. Okay, the word transform just simply means to change. Something in it had been changed by the smooth, dead, smooth strain, right? Something had been changed. Something had been transformed. We were able to then somehow transform this rough strain into being deadly. He didn't know what caused it, but he did know that something had happened. He was not sure what had caused the transformation to happen, but he was 100% sure that a transformation had happened, because otherwise, how would this poor mouse have died? Something was able to change the rough strain bacteria to make it deadly. This is what he ends up calling the transformation principle. So his conclusion is that there is a transformation principle, that there is something that is able to transform non-deadly bacteria, and that something must be genetic in nature. We have these transforming factors, is what he called them. All right. So back to our timeline. So now we have Oswald Avery. Okay. So Oswald Avery comes along in 1943. And at this point, again, we don't know. We know that there are these transforming factors. We just don't know what the heck they are. Most people believe at this point that they have to either be proteins or they have to be nucleic acids. But we don't know what is causing, because again, it has to be one of the four, right? It has to be a biological molecule. So people know it's not carbohydrates. They know it's not lipids. So that pretty much leaves proteins and the nucleic acids. It has to be one of those two. Most people believe that it's actually proteins, because proteins are really, really complex. Like in an average human body, we have something like 150,000 different proteins that do a bunch of stuff in our cells. Avery, however, set up an experiment to prove this. He figured what he actually did was he figured out that nucleic acids, DNA specifically, were the transforming factors, that DNA was the molecule of genetic inheritance. Here's the actual sad thing. He did amazing work, and everybody thought that he fudged his numbers because everyone was so convinced that it had to be proteins. Bad scientists. Anywho. So he said that the transforming factor is this molecule of inheritance, and that this transforming factor had to be DNA. So again, this is science. You can't just say something without actually proving it. So again, how does he go about doing this? All right, so here's his experiment. Oswald basically set up where he had this heat-killed bacteria just like Griffin did, okay? And he added this heat-killed bacteria, he added it to this R strain stain just like Griffin did. And we know that if we combine heat-killed S with live R, that a transformation takes place, which then leads to the death of the mice. Okay, so we know this. So what is causing the transformation? That was his question. So again, there's only 5 macromolecules. Actually, there's 4. There's 4 macromolecules. There's the nucleic acids, RNA and DNA. There's proteins, there's lipids, there's carbohydrates. One of those has to be causing the transformation. So let's see what he did. He took the enzymes that he added enzymes to the heat-killed smooth strain to destroy different macromolecules. So he basically destroyed the RNA on purpose in one test. He destroyed the protein on purpose in a second test. He destroyed the DNA on purpose in the third test. He destroyed the lipid on purpose in the fourth test using enzymes. And he destroyed the carbohydrate on purpose in his 5th test. Okay? So why do this? Well, because if you take out the thing that is causing the transformation, then no transformations should occur. All right, so what was the results? When he took out the RNA, the protein, the lipid, and the carbohydrate, basically there was a transformation that happened, but the mice lived. Because again, we know that adding these two, the mice should, if there is a transformation, they're going to live. But if there isn't one, they're going to die. However, what we saw happen when he destroyed the DNA was that they died, which was kind of crazy. So why? Well, if RNA, protein, lipid, and carbohydrate all have the same result, no transformation, with a transformation, right? And then the DNA, when you take out the DNA, you have death. That's a different result. That tells us that the DNA was responsible for the transformations that occurred previously in the other things. Because when you take out the DNA, you have dead mice. So, what ends up happening? Since DNA prevented the transformation, DNA had to be that transforming factor. So, again, up until this point, we knew that transforming happened. We knew that genetic material got passed from generation to generation. We just didn't know how it happened. Because of Avery's work, we now know that DNA is responsible. But here's kind of the sad thing, no one believed him. Everybody thought he had messed up his numbers because everyone was so convinced it had to be protein. Side note, this is how science works, by the way. Somebody comes up with something so out there that most scientists are like, "Nah, whatever, dude. Like, nope, you obviously messed that up." And it sometimes takes years for other scientists to run experiments to confirm what had happened. So this is what happened to Avery. He came up with something that was so out there, something that nobody was ready to believe, that nobody believed him. And it took almost 10 years of other people running this data for people to actually believe that DNA was the transforming factors and the molecule of genetic inheritance. Back to our timeline, Edwin Chargaff, 1950. Um, we're going to talk more about Edwin Chargaff next week, but in terms of what he did, again, working with DNA because he believed Avery. And he was the minority, by the way. Most people thought it was still protein. But Chargaff, following Avery's work, kept studying DNA, and he found out that in humans, he came up with this thing called the Chargaff rules, where adenine and thymine were always present in equal amount. Guanine and cytosine were always present in equal amounts. So that was what he did. All right, cool. So then comes my personal favorite, Martha Chase and Alfred Hershey. These guys come up with the blender experiments. Literally, they had a food blender in the lab. Like, this was craziness. Anyways, they use bacteriophages, which are literally just viruses that are used to infect bacteria. They use bacteriophages in order to. Oops. What am I looking at? Oh, this is just the wrong one. I'm sorry, guys. I apologize. I have the wrong form open on my other computer because I'm trying to follow along so that you guys can. Oh, silly me. There we go. Awesome. Sorry about that. So these are the blender experiments. So Martha Chase and Alfred Hershey's used bacteriophage, which are viruses that literally infect bacteria. And they do this to determine once and for all what are these genetic inheritance factors. Okay, so they don't have any skin in the game, as it were. They don't know if it's protein, they don't know if it's DNA, but they design an experiment specifically to determine which one of the two is carrying the information. And how they do it is using by bacteriophage. What we know about bacteriophages is that they're very, very simple. They are basically made up of an outer coat of protein, and they have inside of them they have DNA. What we do know happens when a bacteriophage attacks a bacteria is that they will insert their genetic information. So that meant that either this bacteriophage was going to be inserting the protein code into the bacteria, or it meant that it was going to be inserting the DNA into the bacteria. Because we know that when a bacteriophage attacks a cell, attacks a bacteria, it will inject it with its genetic material, hijack the cell, and force the cell to start creating new viruses, which is kind of ingenious from a viral point of view. Okay, so how do they go about proving if it is the protein carrying the genetic material or the DNA carrying the genetic material? All they know is that at the end, the bacteria is going to have this genetic material inside of it. So, using that in order to exclude or test the protein coat, they use bacteria with radioactive 35 sulfur. Proteins have sulfur in them, okay? So they marked it on purpose. They figured if the protein coat was being injected into the bacteria when they first, then they put it into a blender to literally shake the protein shells off of the bacteria, and then they spun it in a centrifuge, which led to the pellet. The pellet is the heavy stuff. The pellet is the bacteria. If the pellet is radioactive, that we know has to contain genetic information. When they ran it with the radioactive protein coats, the radioactivity was only in the liquid, which means it was not in the pellet, which means it was not injected into the bacteria. However, when they infected the viruses with radioactive 32P, radioactive phosphorus, the radioactivity got added to the nucleic acid. So what happened? Same experiment, right? They put in a blender to shake the protein coats off. Um, they spun it in a centrifuge to separate the liquids from the solid pellet. And again, that pellet are the bacteria. They know that the bacteria has to contain the nucleic acid or the genetic information from the bacteriophage. When they ran this experiment, they found phosphorus only in the nucleic acid. They found that trace marker, that 32P, inside the pellet. And again, the pellet is the bacteria. And we knew that whatever this bacteriophage was doing, it had to inject its genetic material into the bacteria. When we find the 32P in the bacteria, we now know that the genetic material had to be from the nucleic acid because that was where the marker was. So, bottom line, what is the conclusion? The conclusion here is beyond a reasonable doubt that DNA is the carrier of genetic information. And the evidence for this, of course, is that they found that DNA in the bacteria, which is where we would expect to find the DNA or the genetic material of the bacteriophage. So that kind of takes us through that. All right. Let me